Dear Vishnu, could you please open your camera and share your screen?
Is still disabled for
Yeah, now we can hear you. Welcome.
Yeah.
Please share your screen.
it's still disabled for share screen.
You cannot share. Is it correct?
Yes.
Okay. Hande, could you please help us?
Hocam.
Merhaba, nasılsın Hande?
İyi, çok teşekkür ederim. Bir şeyle ilgileniyordum da bir
Tamam,
şeyin
ekran paylaşımı, Vishnu ekran paylaşımı yapamıyor şu anda.
Tamam. Hocam hemen. Tamamdır.
Teşekkürler.
Oldu mu? Oldu galiba.
Can you do it now? Please try it again. Okay, perfect.
Can you see it?
Yeah, it is visible now. If you are ready, you may start.
Yeah. Okay. Assalamualaikum warahmatullahi wabarakatuh. Let me introduce myself. My name is Wisnu Rizkulazufar. I come from Indonesia. And here I will present my research with title.
Can you
Potential
talk, please, loudly a little bit?
Okay. This is my research. It is about with title Potential of Chanchapidra or Philanthus neruri as an alternative anti-inflammatory with a mechanism for inhibiting TNF-alpha and ERK2 activity in silico. So yeah, in background, let us to review about inflammatory mechanism. When a wound occurs on the skin, several pathogens will enter the body, and then pro-inflammatory cytokines cause monocyte and neutrophils to do phagocytosis process. But if this wound is not handled properly, it will cause raised complication, including diabetic complication, various cancer, cardiovascular disease, wound joint, autoimmune disorder, metabolic and endocrine disorder, pulmonary disease, neurological disorder, and more things again. In other hand, our background is the magnitude of the influence of inflammation on disease, the presence of side effects, and the availability of potent anti-inflammatory drugs for long-term use. Safe alternatives to anti-inflammatory drugs are needed, and utilizing Indonesia's biodiversity. And in first studies show that conventional drugs such as diclofenac have minor side effects such as back pain and headache. Apart from that, these synthesis drugs also have minor side effects such as bleeding and respiratory failure. Therefore, some synthetic drugs cannot be used for chronic disease with long-term drug use. In contrast to synthetic medicines, although herbal medicine also have side effects too, they can still be tolerated by the body, so they are safer when used for a long time. And this is our herb. This is what is it? Chanchapedra or Philanthus neruri. In basic theory, when a pathogen enters the body, it will be recognized by toll-like receptor 4 in here, and then which will then activate a secondary cascade, the main of which is ERK2 in ERK complex. Sorry. And then after that, it will initiate to produce pro-inflammatory agents or cytokines like TNF alpha and etc. By synthesis protein mechanism. On the other hand, TNF alpha it will initiate ERK complex to express pro-inflammatory agents like FCAM1 and CHOX2 and etc. In previous research, it was known that Chancha-Pedra or Philanthus neururi is able to inhibit the expression or activity of TNF alpha. However, this research was directed at inactivating that secreted TNF alpha protein. The secreted TNF alpha has the ability to initiate ERK2 expression to produce pro-inflammatory mediators in the protein synthesis process. On the other hand, ERK2 is inhibited in order to reduce the production of pro-inflammatory mediators so that an immunosuppressant effect can occur. The aim of this research is an effort to develop drug discovery, where research will be carried out to look for the same structure or structural relationship of herbal active compounds with native ligands or control so that they can cause certain effects. In this study, the dessert effects was anti-inflammatory. After obtaining a suitable candidate, the structure of the active herbal compound will be imitated to be synthesized for the mass creation of new drugs with several side effects. How about the method? This research was carried out with a computational design using the molecular docking method. Apart from that, this research also used the PKCSM online database to determine the physicochemical and pharmacokinetic properties of the active compound of Philanthus neururi. It also carried out by attaching the active compound of Philanthus neururi to target proteins, such as tumor necrosis factor and extracellular signal regulated to or ERK2 using the autodocking application. And then this is our control with TNF alpha and ERK2 with each other interaction with native ligand. And this is our results. From the result of molecular docking of TNF alpha, correlagin is the best active compound because the free bound energy value is smaller than -7, and there is an amino acid residue similarity of 25%. And then in ERK2, from the result, correlagin and quercetin are the best active compounds because the free bound energy value of correlagin is smallest, and quercetin has the best presentation of amino acid residual similarities. And then because the molecular docking result show that the best compounds are correlagin and quercetin, the pharmacokinetic profile results only focus on correlagin and quercetin. Quercetin has a good absorption profile, namely greater than 70% in here. And it's not hepatotoxic and cytotoxic in profile of toxicity. Quercetin is not hepatotoxic and cytotoxic. However, the absorption of correlagin is relatively low because less than 60%, yeah, in the human intestine, but it is not hepatotoxic and cytotoxic in toxicity profile. In our conclusion, correlagin has affinity for TNF alpha and ERK2, while quercetin only has affinity for ERK2. Correlagin has low solubility so that the absorption process in the intestine is slow, while quercetin has good solubility. Quercetin has good pharmacokinetics, is safe and non-toxic. Correlagin has low bioavailability when given orally, but is not toxic. quercetin can be given as an oral preparation, but coraligin is more recommended as a parenteral preparation. And this is our references. Thank you.
Thank you very much for the nice presentation. Dear participants, do you have any questions? Professor Aykaç? You may have one.
Yeah, but everything is clear, and to be honest, it's a little bit far away from my professional field. Not so much, a little bit. We are working in a little bit more specific content and topics nowadays. I used to work on the targeted drug delivery system based on the anti-cancer studies, but nowadays we are shaping our scientific research mainly on organic waste material based nanomaterials that are useful for biomedical application. That's why a little bit, kind of slightly, I am far away, and with the connection that I don't have any kind of question so far. And by the way, I am just wondering about something personal. Who is Dimitri Damayanti? Is your supervisor?
Yes, please.
He or she, I don't recognize, and I don't want to make any mistake.
She is my supervisor.
She's your supervisor. Congratulations. She's putting so much effort and so much presentation here, as far as I see.
Yeah,
If
yeah.
she's around, I would like to say hi to her as well. Congratulations. Too many presentations. Well done. By the way, where are you from?
I come from Indonesia, Professor Ahmed. I come from Islamic University of Malang in East Java, Indonesia.
Okay, now are you in Malaysia?
No, I'm in Indonesia.
In
Indonesia.
Indonesia.
Indonesia. Now you are over there, right?
Yeah, I'm here. I'm
What
in
time is
Indonesia.
it over there?
In 3 p.m. sir.
3 p.m. Okay, okay. Congratulations to you and to your supervisor. Well done.
Yeah.
I wish you all the best.
Thank you. In here, I have two friends with same supervisor and same university too.
Okay, that's great. That's great. I can see from the schedule and from the program, and I see the name of the Damayanti very frequently, and I realize that you have so many study. I'm looking forward to listening the other one as well. You have one,
Yeah.
2, 3, at least 3 I saw, maybe even more, 4, maybe.
Just 3 in here in this season.
By the way, glad to see you, Professor Özger.
Thank you very much.
Dear Faruk. Faruk is my one of the good friend, and
Thank you.
we've been know each other for a long time, for many years. That's why I take this opportunity to saying hi to you as well.
Thank you. I also greet you.
Thank you.
Nice to see you again.
The same. I'm very glad to see you.
Thank you for your nice remarks and comments also. Dear Vishnu, thank you very much for your nice presentation again. And we are ready for the next presenter, Ibrahim Karidio. Is it correct? I think you will present the research. Is it correct, Ibrahim?
Good morning, yes, the name is correct, but I'm not the presenter. The presenter is François Comlan Aida. I think she's connected, I think.
She's connecting now. Is it correct?
Okay,
Is she here?
okay. I think she's yes, she is connecting now.
Okay,
She's
okay, let's
connecting.
wait. Let's wait.
Mm.
You are the co-author of the study, Ibrahim?
Yes, actually, I'm her mentor. She works in my lab. She's one of my students.
Okay, thank you. From which university? From where?
She's regularly registered at the University Abdou Moumouni of Niamey, that's Niger Republic. And she's doing her work here at CERMES, that's a National Research Center, Medical and Public Health Research Center. She's doing basically all of her lab work here in CERMES, and she's also working here. But regularly, she is registered at Abdou Moumouni University. That's one of our public university in Niger Republic, not Nigeria. Niger and Nigeria are neighboring
different
countries,
countries.
but
Yes,
yeah.
I know. I know
So
it.
maybe if she's not able to connect right now, maybe you can just move on to the next one,
and she can present later at the end of the session.
Yes.
Okay.
Yes, thank you.
Yeah. You are welcome. So the third presenter is, let me check the name, Nuris. Yes, Hidayah. Can you hear me, Hidayah?
Yes, I can hear you.
Okay. Could you please share your screen? Welcome. From the same university,
Yes.
with the same advisor. Dear Hidayah, if you are ready, please start. And before starting, please make your... Okay, this is very good.
Okay. Assalamualaikum warahmatullahi wabarakatuh. It's a pleasure to have the opportunity to speak before you today. Let me introduce myself. My name is Nuris Bashirotul Hidayah, and I'm from Islamic University of Malang, Indonesia. Today, I'm going to present an abstract with title Antibacterial Effect of Aloe Vera Ethanol Extract Against the Growth of Bacteria Staphylococcus aureus and Escherichia coli through Zoe test, MIC, and MBC measurement. Introduction. Aloe vera is known to have active compounds that act as antibacterials. Staphylococcus aureus is a gram-positive bacterium, while Escherichia coli is a gram-negative bacterium that often causes infection, so it requires antibiotic treatment. The use of antibiotics can cause resistance. Until now, there have been no studies that assess the effect of aloe vera ethanol extract on the inhibition of growth and death of Staphylococcus aureus and Escherichia coli bacteria, so it needs to be studied. Materials and methods. Preparation of aloe vera ethanol extract by maceration method. Furthermore, phytochemical tests are carried out to determine the active compounds in the extract used. Zone of inhibition test was performed by well diffusion method using different concentrations, control positive and control negative. The test to determine the minimum inhibition concentration or MIC and minimum bactericidal concentration or MBC use liquid and solid dilution methods using different concentration. For research methods, first preparation of aloe vera ethanol extract. At first, simplicity of aloe vera soak with 70% ethanol solvent and then filter with vacuum Buchner and then evaporated using a rotatory evaporator and then dried using oven. Phytochemical testing was performed on aloe vera ethanol extract, then put into a spirit funnel filled with a mixture of chloroform and ethanol 70 solvent. Then leaves it in the split funnel until 2 layers of chloroform and filtrate are formed. The filtered phase is used to test flavonoids, phenolics, and saponins, while the chloroform phase is used to test for alkaloids and terpenoids. And third, zone of inhibition test at first is preparation bacteria, and then inoculation into Muller-Hinton agar. After that, whole and inserted stock. After that, incubate for 24 hours, and the test was repeated 3 times. And then diameter measurement, the bright zone. MIC test, determination of variable concentration, and then standardization with standard McFarland, and then the bacteria was inoculated into Muller-Hinton broth. After that, testing MIC. Prepare a test tube and fill with Muller-Hinton broth, and then incubate for 24 hours. And then determination MIC. The test is repeated until the same MIC value is obtained 3 times. MBC test, at first is determination of concentration, and then prepare nutrient agar that divided into portion 6 parts. After that, streaking in one section, and the test was repeated 3 times. After that, incubate for 24 hours and determination MBC. For data analysis, the phytochemical tests of aloe vera ethanol extract were analyzed descriptively, and zone of inhibition or ZOI test data were tested for normality and homogeneity to determine normal distributed data. After the data was normally distributed, P more than 0.05 and homogeneous, then one-way analysis of variant test was carried out. All the statistical needs carried out using the statistical package for the social science or SPSS version 22. The result of minimum inhibition concentration or MIC and minimum bactericidal concentration or MBC are described combined by supporting photo. Results, the studies aloe vera ethanol extract contain flavonoids and alkaloids. The measurement results of the inhibitory zone at concentration less than 50% were 0 millimeter. At a concentration of 50%, it was 7.67 millimeter. At 100% concentration, it was 20.67 millimeter, while in positive control tetracycline and amoxicillin were 34 millimeter and 36 millimeter. Minimum inhibition concentration or MIC and minimum bactericidal concentration or MBC of aloe vera ethanol extract against Staphylococcus aureus at a concentration of 12.5%, while against Escherichia coli at a concentration of 25%. These are the picture of the results. Picture 1, result of ZOI test Staphylococcus aureus, and picture 2, result of ZOI test Escherichia coli. The zone of inhibition or ZOI is a circular area around the antibacterial site where bacterial colonies don't grow. This zone of inhibition is used to measure the susceptibility of bacteria to antibacterial substance. If the antibacterial agent effectively inhibits bacterial growth, there is a clear zone. The measurement can be done using a ruler or a caliper. Picture 3, result of MIC and MBC measurement, Staphylococcus aureus, and picture 4, result of MIC and MBC measurement, Escherichia coli. MIC can be defined as a technique to determine the minimum concentration of an antibacterial agent required to inhibit bacterial growth. Bacterial growth can be seen from the media that becomes cloudy or there is a sediment demand. And while MBC is the minimum concentration of extract that can kill bacteria, determination of MBC value is seen from the absence of bacterial growth on the streak plate results, which indicates that each test bacterium dies because the test solution at that concentrations. Conclusion, aloe vera ethanol extract has antibacterial effects and is able to inhibit and kill Staphylococcus aureus and Escherichia coli bacteria with higher inhibitory power against Staphylococcus aureus than against Escherichia coli. These are the references of my study. Thank you for the time and attention.
Thank you very much, dear Hidayah, for the nice presentation again. Dear participants, is there any question or comment?
Yeah, congratulations. I don't know what to say. Actually, I am quite interested about the result, but at the same time, there is a kind of a different way of thinking. You are taking the ethanol extract of the aloe vera. I mean, the alcohol extract. What does it mean? It means that we all know that alcohol has a killing effect on many different types of bacteria itself. So that effect comes from aloe vera extract, or that effects come from the ethanol. The extraction is important. If it was water-based extraction, it could be different result maybe. Have you tried to continue with the water-based extract? This is the first question. And the second question, how you support your scientific background or scientific outcomes of this study that comes from the ethanol extract, not from the water extract? Do you understand my question? I can make it a little bit shorter and more clear for you. Because first I make the comment and then I try to ask you the question. Maybe you're confused. So without any comment, the point is that, have you tried that aloe vera extract, the water extract, water-based extraction, to follow the same procedure and to see if there is any kind of effect against those bacteria or not?
Okay, thank you for the question. I will answer the question. So in previous research by Azahra et al. In 2019, aloe vera extract with aqua dissolvent was able to provide antibacterial effect. And then in other studies, extraction using ethanol solvent produce higher extracts produce. The higher the content of substance or active compounds that are attracted.
Yeah, you telling me that another study were carried out with aqueous extract and they presented that it has an antibacterial effect, right? Yes. So in your case, the ethanol extract seems, according to your idea, according to your argument, you say since it gets more ingredients from the aloe vera, so it has a more active side and the activity against this bacteria is higher, right?
Daya, is it correct? We cannot hear you now. Could you please switch on your microphone?
Okay. And in other studies, in my maceration method to extract aloe vera ethanol extract is drying. This is to remove 70% ethanol content from the extract. So the extract is remove 70% ethanol.
Okay. If your supervisor over there, I would like to talk to her actually, because this is scientific symposium, and I hope that to have a kind of meeting or collaboration or common study together. If she's around there, I would like to ask her some question. To be honest, we are working on the same field, more or less, and we are nowadays trying to get. We cannot hear you.
Dear Hidayah. Okay. Hidayah?
To attack the other resource, extraction using ethanol solvent produces higher extract, the higher the content of substance or active compounds.
Yes, I understand. You are claiming that the amount of the active substances is increasing when you perform the ethanol extract. Yes, I agree with you. I understand you. But still you have to eliminate the alcohol effect. You have to apply ethanol itself as an Bayer study, as a control experiment. That's why I asked if your supervisor around, let us to meet and to talk separately because we don't want to ruin this excellent symposium because we have to be on time as well. If she's over there or she's around, please let her to know my email address at least and to get in touch with me, please.
Okay, Prof, thank you.
Professor Aykaç, could you please send your email address to the chat box and Hidayah can save it? And
Of course, thank you.
yeah,
I will
it
do
will be
that.
more helpful. Thank you very much for your contribution.
Thank you.
Dear Hidayah, thank you very much. Dear participants, do you have any other comment or remark? If not, we have the next presenter. Thank you again, Hidayah. And the next presenter is Teodora Kukric. Kukric? Dear Kukric,
Yes,
welcome. Welcome, hello.
thank you.
Nice
Just
to
a
see
second.
you. Okay, please share your screen.
Can you see it now?
Yeah, it is perfect. You may start if you are ready.
Thank you. My name is Teodora Kukric, and I'm a microbiologist at Faculty of Agriculture in University of Novi Sad in Serbia. And my team members and I are investigating natural deep eutectic solvents. So today I will present our work, the environmental aspect and biological activity of natural deep eutectic solvents. Natural deep eutectic solvents are abbreviated as NADES. And since the beginning of the 21st century, they have been extensively researched in different scientific fields, and they developed as an alternative to toxic and synthetic chemical agents. To promote green and sustainable chemistry. Demand for that were environmental preservation and prevention of harmful effects caused by, as I said, toxic chemical agents. In the past 2 decades, NADS have undergone a scientific evaluation and have been incorporated in a few industries because their multifunctionality, and they are utilized in chemical, pharmaceutical, and food industries as solvents or co-solvents. They are used in extraction process or as catalyst in synthetic processes. And NADS, so they represent a new type of solvents, and they are a result of research efforts for non-toxic, non-flammable, and less volatile solvents. And they are switch for mainly use organic solvents, and they are environmentally sustainable, and they are connected with biodegradability. As you can see in the picture, NADES or NADS are a combination of two or more components, which can be either solid or liquid. And when these components are mixed in a specific proportion or molar ratio, their melting point significantly decreases, causing them to become liquid at room temperature. As you can see in the picture, there is component 1 and component 2, and in the middle is the NADS in a liquid state, and it will remain in that liquid state at room temperature. And mostly compounds that are constitute the NADS are primary metabolites, amino acids or organic acids, sugars or choline derivatives. And those are called hydrogen bond donors and hydrogen bond acceptors, as you can see at the top. Those components are hydrophilic in their nature, and hydrophobic deep eutectic solvents, abbreviated as HDES, are a subclass of NADS, and they consisting of compounds such as terpenoids and fatty acids, which are hydrophobic and volatile compounds, and some of terpenoid antimicrobial activity is or was proven in previous publications. So studying HDS for antimicrobial purposes offers the benefit of including a wide range of components in their composition. So terpenoids-based HDS represent the solvents that theoretically have the potential to be used as antimicrobial agents. And some of that terpenoids are menthol, thymol, camphor. So utilization of HDS has led to research on their biological properties, such as antimicrobial activity, because it is required verification from environmental sustainable point of view. So this work aimed to examine the antimicrobial activity of one HDS from menthol and lactic acids, and due to HDS adjustable nature, so they can be tailor-made, and the knowledge of previous publication of antimicrobial activity of menthol and lactic acids, it is hypothesized that HDS formulated by combining menthol and lactic acids can exhibit antibacterial activity. Nowadays, it is challenging for humankind to find environmentally friendly but toxic agents for pathogenic microorganisms, and antibacterial resistance is also a growing problem, so alternative solution is needed. And the hypothesis is that menthol lactic acids has antimicrobial activity. First, antibacterial activity against human pathogen in Latin Escherichia coli, and second, antifungal activity against phytopathogen Botrytis cinerea. And also that menthol lactic acids is not phytotoxic. The antibacterial activity or antibacterial assay is conducted by microdilution method, using twofold dilutions in a concentrational range 0.25 to 25%. And after 24 hours of incubation, TTC indicator was used for visualization of minimum inhibitory concentration, which is 3.13% concentration of HDS. And antifungal activity or antifungal assay was conducted by agar diffusion method. And also twofold dilution of HDS were used in concentrational range 0.7 to 100% or undiluted HDS. After 7-day incubation, visual observation was followed by measuring diameter of mycelial growth or botrytis colonies, and then relative inhibition of mycelial growth was calculated, and you can see in the picture a result of agar diffusion method, and where at 0.7 concentration of HDS, 85% inhibition, and when 100% or undiluted HDS was applied, there is no fungal growth, or there is a complete inhibition of fungal growth. While phytotoxicity assay is conducted on tuber potato slice in the same concentrational range of HDS as in antifungal assay from 0.7 to undiluted HDS, 10 microliters of each concentrations was applied in the center of tuber potato slice, and after 24 hours, visual changes are observed, and menthol lactic acid exhibited phytotoxicity at 50% and 100%. So conclusions of our research study are that microorganisms or different pathogens respond differently to HDS, and that menthol lactic acid has the potential as an antimicrobial agent, and could be used to control infectious disease caused by these microbial pathogens. But even though that potential and this preliminary testing, more studies are required or recommended to define the impacts of HDS, specifically menthol lactic acids, HDS. Thank you for your attention.
Thank you very much, Theodora, for the useful, nice presentation. Dear participants, any comments, any idea or question? If yes, we have 2 minutes. Otherwise, I will pass to the next presenter. No? Okay, dear Theodora, thank you very much again for the nice presentation.
Thank you.
I hope the best for you.
You too. Goodbye.
Bye-bye. Please stay here. At the end of the session, we will have a photo.
Sure,
So
okay.
the next presenter is Alfia Salsabil.
Yes,
Okay. Hello, welcome, Alfia.
yes, hello.
Hello.
Hey, can you see my
Yes, we can see it now.
presentation?
Yeah, we can. It is visible.
Okay, good afternoon, everyone. Thank you all for being here today. My name is Alfia Salsabil, and I'm from Indonesia at University of Islam Malang. Today, I want to presentation about hyperglycemia and pancreatic damage in sucrose immersion induced zebrafish. Diabetes mellitus, especially type 2 DM, is a metabolic disease characterized by hyperglycemia and structural changes in the Langerhans islet and a decrease in the number of pancreatic beta cell. Zebrafish is one of the animals that can be used as a model of hyperglycemia. The study aims to prove the effect of sucrose immersion on blood glucose levels and changes in Langerhans islet structure in zebrafish. For materials and methods, laboratory research using post-test group control only research design. Sample is 30 adult male zebrafish. The research group consists of N is normal, G1 is 1% sucrose immersion, G2 is 2% sucrose immersion, and G3 is 4% sucrose immersion. Sucrose induction was carried out for 28 days. Water changes are carried out every 2 days. The sacrifice process was carried out by placing the zebrafish in ice water for 10 minutes. After the fish unconscious, the tail was cut to check fasting blood glucose level using glucotest. Next, the pancreas organ was taken. Pancreatic organs were made in the form of prepared histology with hematoxylin and eosin staining, counted the number of cells experiencing necrosis and the area of Langerhans islet. Data analysis using ANOVA with significance is p less than 0.05. The result and conclusion, the highest glucose level was achieved at G3 is 188.67 plus minus 15.97 milligram per deciliter, which was different from normal G1 and G2. The level of Langerhans islet damage was highest in G3 is 81.8% compared to normal is 3.8%. G1 is 33.8% and G2 is 62.65%. The diameter of Langerhans islet was smaller in G3 is 69.698 plus minus 8.47 micrometer compared to normal G1 and G2. That is, can be concluded that 4% glucose induction causes an increase in blood glucose levels and Langerhans islet damage. And this is the result of blood sugar examination of zebrafish from 6 samples. Normal with the mean is 84.17 and standard deviation is 5.71, is 16%. And from G1 with the mean is 131.67 and standard deviation 3.68 is 24%. And G2 is with the average 137.83 and standard deviation 4.71 is 25%. And G3 with the mean is 188.67 and standard deviation 15.97 is 35%. And this is the histopathology of zebrafish pancreas magnification 100 times. A is normal, B is 1% sucrose immersion, C is 2% sucrose immersion, and D is 4% sucrose immersion. The circle part is the islet of Langerhans. This is the histopathology of zebrafish pancreas magnification 400 times. The arrow yellow is pycnotic, the arrow blue is karyorrhexis, the arrow green is karyolysis, and the arrow black is empty space between cells. This is the table of percentage of pancreatic Langerhans islet damage. G1 with a mean is 1.23, with the percent damage is 33.8%, indicating mild damage. G2 with mean 2.38, with percentage damage is 62.65%, indicating moderate damage. G3 with the mean is 3.15, with percent damage is 81.8%, indicating severe damage. And normal is 0.05 with percent damage is 3.8%, indicating normal. And this is the table of main Langerhans islet diameter. from group G1 is 99.292 plus or minus 4.34 micrometers, and G2 is 86.255 plus or minus 5.80, and G3 is 69.698 plus or minus 8.47, and normal is 122.096 plus or minus 16.82. And this is the reference used. That's everything I want to say about my presentation today. Thank you for your time and attention.
Thank you very much, dear Alfia, for the nice presentation. Dear participants, any comments? Any question? Okay. Thank you again, Alfia.
Thank you.
I wish the best for you. And the next presenter is Şule Nazlı or Mehmet Şulha. Şule, you are here.
Evet,
Okay.
buradayım hocam.
Tamam. İngilizce mi yapacaksın, Türkçe mi?
Türkçe şeklinde sunacağım hocam.
Türkçe, tamam.
Ekran kaydını.
So this presentation will be in Turkish. After this presentation, we have one presenter, Aida, and we will share a video of Aida at the end of this session. Please stay here, don't leave. Başlayabilirsin hazır olduğunda.
Merhabalar herkese. Ben Şule Nazlı Ergün. İzmir Kağıt Çelebi Üniversitesi Su Ürünleri Fakültesi'nde yüksek lisans öğrencisiyim. Araştırma konum İzmir İç Körfezi'nde dağılım gösteren mollusk faunası. İlk olarak genel bir giriş yapmak istiyorum konuyla ilgili. Yerküre karasal, denizler ve tatlı su olmak üzere başlıca 3 ekosisteme sahiptir. Bilimsel çalışmalarla okyanus ve denizlerde yaklaşık 200 bin canlı türünün bulunduğu saptanmış olup bunların büyük bir kısmının dipte yaşayan bentik canlılar adını verdiğimiz organizmalardır. Denizler ekosistemi içinde yer alan bentik bölge bu büyük ekosistemin esasını oluşturmaktadır. Sahil çizgisinden başlayarak en derin deniz diplerine kadar uzanan deniz tabanını kapsamaktadır ve derinliğe bağlı değişen çevresel koşullar ve dip yapıları bentik fauna ve floranın dağılımını belirleyen en önemli faktörlerdendir. Bentik bölge adını verdiğimiz deniz dipleri yumuşak ve sert substratum gibi pek çok farklı yapılarda olabilmektedir. Kaya, kum, çakıl ve çamur gibi. Görsellerde gördüğümüz üstteki iki kare yumuşak substratum örnekleri, balçık ve çamur, kumlu çamur daha çok. Altta gördüğümüz midye fasulyesleri de sert substratum örneklerini oluşturmaktadır. Ve birçok deniz canlısının tercih ettiği yaşam ortamıdır aynı zamanda. Bentik bölgede yaşayan ve yumuşakça olarak isimlendirdiğimiz molluska filumi buldukları ortamlara iyi adapte olabilmektedirler. Yine sucul ve karasal olarak dağılım gösterebilmektedirler. Tüm bu tür zenginliğinin yanı sıra çok sayıda ekonomik türde kapsaması nedeniyle insanların en fazla ilgisine çekmiş bir omurgasız filumudur. Yine görselde gördüğümüz aynı zamanda ekonomik tür olan ve avcılığı ve yetiştiriciliği yapılan Akdeniz midyesi adını verdiğimiz türümüz. Molluska türleri canlılar arasında içerdiği tür ve birey sayısı bakımından önemli bir yere sahiptir ve çeşitli kaynaklara göre değişmekle birlikte denizlerimizde, dünya denizlerimizde 52 bin kadar türü bilinmektedir. Yumuşakça adını da verdiğimiz molluska filumi toplam 8 klasis altında incelenmektedir. Görsellerde belirttiğim ilk klasis Caudophyllata klasisi, ikinci klasis Solanagastres klasisi, Monoplacofora klasisleri, Polyplacofora klasisleri, deniz dişlerine benzettiğimiz Scapopoda, ahtapotların olduğu grup Cephalopoda grupları ve Gastropoda ve Bivalvia sınıfları. Araştırma konumuz daha çok gastropod ve bivalvler üzerine yoğunlaşmaktadır. Bu sınıflar içinde tür sayısı bakımından en zengin sınıf gastropoda sınıfı olup Akdeniz ekosisteminde 1500 civarında türü bilinmekte ve bunlardan 422'si bivalvia sınıfına aittir. Yine ülkemiz denizlerine yapılan checkliste, kontrol listesinde 1065 tane molluska türü bildirilmiş olup bunlardan 706 tanesinin gastropoda sınıfı kapsamında olduğu, 279 tanesinin ise bivalvia sınıfında kapsamında olduğu bilinmektedir ve bu sebeple daha çok tür sayısı bu sınıflara ait olduğu için araştırma konumuzda gastropoda ve bivalvia sınıfına yoğunlaşılmıştır. Denizlerimizde yapılmış bentik çalışmalar Ege Denizi'nde 1970'li yıllarda yoğunluk kazanmış olup araştırmanın konusunda içine alan türlerin de dahil olduğu Moska fanusu ile ilgili yapılmış çalışmalar Forbes tarafından gerçekleşmiş olup günümüze kadar pek çok araştırmacı sayılı çalışma yürütmüştür. Bu bentik organizmaların tür kompozisyonunu, tür çeşitliliğini, biyomas değerlerini, ekolojik yapı ve su kalitesini, kirliliği belirlemede de önemli indikatörler olarak kabul edilmektedir. Dip organizmalarından özellikle molluska türleri dip çamurunu havalandırmakta ve mineralizasyonu önemli derecede etkilemektedir. Genel bir girişten sonra çalışmanın materyal ve yönteminden bahsetmek istiyorum. İzmir İç Körfezi'de doğum gösteren mollusk fanusunu belirlemek amacıyla araştırma bölgesinden belirlenen 6 istasyondan Temmuz 2023 ve Mart 2024 tarihleri arasında örneklemeler gerçekleştirdim. Burada sırayla istasyon bilgilerimi görmekteyiz. İncelenen materyal 0,5 ve 6,5 metre derinlikler arasından yine yumuşak ve sert substratım olmak üzere çeşitli biyotoplardan el grabı, kuadrat ve spatula adını verdiğimiz yöntemlerle gerçekleştirilmiştir. Görselde Van Wien grab adını verdiğimiz örnekleme materyalimiz ve spatula ve kuadrat örnekleme yöntemimiz. Sert substratım için kuadrat kullanıyoruz. Bu substratlardan toplanan örnekler 5 litrelik bidonlarda görselde gördüğümüz gibi yerleştirilip üzerine formaldehit ilave edilerek laboratuvarda incelemeye hazır hale getirildi. Laboratuvara getirdiğimiz örnekleri hafif basınçlı su altında 0,5'lik ilk görselde gördüğümüz gibi elek sistemiyle yıkayıp incelenmek üzere hazırlıyoruz. Sonrasında %70'lik etil alkol bulunan kaplara aşağıdaki görseldeki gibi ayırıyoruz. Ve bunu takiben makro ve mikro boyutta ayrı ayrı türleri gruplarına ayırdıktan sonra binoküler mikroskop yardımıyla gastropoda ve bivalvia türlerinin tayinlerini yaparak tespitlerini gerçekleştirdim ve birey sayılarını belirledim. Tür tayinlerini genellikle kabuk özelliklerine göre gerçekleştirdim. Bunu da yine belirlenen çalışmalardan, checklist'ten, Clemon ve Worms adını verdiğimiz çalışmalardan dikkate alarak tür tayinlerini gerçekleştirdim. İstasyonlara ait genel özellikler bu şekilde. Her bir istasyona ait koordinatlar, örnekleme tarihleri, derinlik bilgileri, özellikle biyotop bilgileri bizim için çok önemli ve örnekleme araçlarına dair tabloda gördüğünüz gibi belirledim. Kısaca istasyonların genel özelliklerinden bahsetmek istiyorum. İlk istasyon bölgem İnciraltı. Bu örneklemeler İzmir Kayıkları Yelken ve Konu Tesisi'nden gerçekleştirildi. Tip yapısı çoğunlukla kırık deniz kabuklarının yer aldığı kalkalı kumlu çamur şeklinde 0,5'e 2 metre derinliklerden gerçekleştirdik. Ve bölgenin görselleri gördüğünüz gibi. 2 nolu istasyon Vapur İskele. Burada dip yapısı daha çok iri çakıllı, taşlı ve kumlu bir bölge. Balıkçı teknelerinin ve vapurlarının bulunduğu uzun bir sahil çizgisinden oluşmakta. Gözlemlerimize göre bölgede kayaların üzerinde çok sayıda Akdeniz midyesi türü, ikinci fotoğrafta görüyoruz, mevcut. 3 nolu istasyon Pasaport İskele'nin bulunduğu Kantar Polis Karakolu. Burada da yine dip yapısı benzer kalkulu kumlu çamur şeklinde. Ve sert substrat örneklerini topladığımız lastikler ve halatlar üzerindeki midye fasiyerslerini gözlemledim. 4 nolu istasyon liman bölgesi. İzmir Denizdibi Tarama Baş Mühendisliği bölgesinden seçilmiştir bu istasyon. Körfez'in önemli bir bölümünü oluşturmaktadır. Yine dip yapısı burada gördüğümüz gibi balçık, çamur, siyah bir balçık çamurdan oluşmakta. Kıyı bölgeleri de şu görseldeki gibi büyük bir kayalarla kaplıdır. Yine üzerinde çok sayıda Akdeniz midyesinin bulunduğu. 5 nolu istasyon Bayraklı. Yine benzer özellik gösterir dip yapısı olarak balçık çamur, yer yer midye fasiyesleri bulunmakta, büyük taş ve kayalar bulunmakta. Gözlemlerimize göre kıyı balıkçılığının yapıldığı bir bölge olduğu için atık maddelerin suya düşmesi ve av malzemelerinden dolayı yoğun bir bölgede kirlilik görünmekte. Çevresel bir kirlilik. 6 nolu istasyon Yelken Kulübü. Karşı Yelken Kulübü'nün olduğu bölgeden seçtiğimiz bu istasyonun dip yapısı yine kırık deniz kabuklarının bulunduğu kalkalı ve kumlu çamurdur. Kıyı bölgelerinde bu üst resimde gördüğümüz gibi ve yandaki resimde bol miktarda Akdeniz midyesi mevcuttur. Çalışmam hala devam etmekte ama sempozyum tarihine kadar ancak sonbahar ve kış örneklerinin tayinlerini gerçekleştirebildim. Halen yaz ve ilkbahar örneklerinin tayinlerini sürdürüyorum. Ve tüm bu araştırmalar sonucunda bugün şu ana kadar bulduğum taksonomik bulgular şu şekilde. Yaklaşık 6 istasyondan 5 ordo, toplamda 8 familya, 12 tür ve 79 birey tespit ettim. Sonbahar ve kış mevsimine ait tespit ettiğim türlerin istasyonlara göre tür ve birey sayılarını görmekteyiz. Yine Gastropoda sınıfı için ayrı bir tablo. Bivalvia sınıfı için toplamda 3 farklı tür mevcut ve bunların sayıları. Bölgede tespit edilen türlerin familya ve tür birey sayısı olarak ayrı tablolar hazırladım. İlk olarak ikişer tür sayısı ile Fissurellide familyası, Ceritide familyası, Muricide ve Nasseride familyaları ilk 4 sırada yer almakta ve bunları da birer tür ile Trochide familyası, Arkide, Anomide ve Veneride familyaları temsil etmektedir. Toplamda 8 familya ve 12 tür tespit ettik. Birey sayısı olarak baktığımızda ise yine en yüksek birey sayısı Nasseride familyasına ait 27 bireyle. Bunu Ceritide familyası 21 bireyle takip etmekte. Muricide familyası 10 birey, Trochide familyası 8 birey, Veneride familyalarına ait 7 birey, Fissurellide familyasına ait 3 birey, Anomide familyasına ait 2 ve Arkide familyasına ait 1 birey tespit ettim. Tespit ettiğim türleri görmemiz adına taksonomilerini ve görsellerini ekledim. Yine Diodora gibberella adını verdiğimiz bir gastropod türü. Sonrasında Fissurella nubecula türü, yine gastropod klasisinde. Sonrasında Gibbula adansoni adını verdiğimiz Trochide familyasına ait diğer bir tür. Yine Ceritide familyasında iki farklı tür vardı. Bunlardan biri Bittium reticulatum, görselde gördüğümüz tür. Bir diğeri ise Seritium scabridum adını verdiğimiz bir diğer gastropod türü. Muricide familyasına ait yine 2 tür söylemiştik. Hexaplex trunculus, görselde gördüğümüz türlerden. Nasseride familyasına ait Nassarius corniculum türü ve Nassarius reticulatus türü, 2 farklı tür tespit etmiştim. Son olarak Bivalvia klasisine ait de 3 farklı familya ve tür vardı. Bunlardan Arkide familyasına ait Anadara transversa türü, bir bival türü, yani bir çift kabuklu. Yine Anamio epiphyum adını verdiğimiz yine bir çift kabuklu bival türü. Ve son olarak Veneride adını verdiğimiz Veneride familyasına ait olan Rudisapes texuatus türünü görselde bu şekilde görmekteyiz. Sonuç olarak İzmir İç Körfezi'de belirlediğimiz 6 istasyondan 0,5 ve 6,5 metre derinlikler arasında sert ve yumuşak substratım olmak üzere farklı biyotoplardan örnekler yaptım. Ve bu değerlendirmeler sonucunda sonbahar ve kış mevsimine ait türlerin tayinlerini gerçekleştirerek bulgularda bu şekilde verdim. 5 orada 8 familya ve 12 tür, 79 birey tespit ettim. Türkiye kıyıları boyunca molluska çeşitliliğini ortaya koymak için hazırlanan kontrol listesinde buradaki 1065 tür bildirilmişti ve 279 tanesi bivalvia, 706 tanesi gastropoda sınıfındaydı. Tayin anahtarı olarak bu checkliste konuyla ilgili diğer çalışmalara dikkate aldığımda büyük çoğunluğunun kıyılarımızda bilinen ve bol bulunan türler olduğunu tespit ettim. Tespit edilen türlerin düşük sayıda olması, araştırma bölgesinin yalnızca kıyısal ekosistemden, yani 0,5 ve 6 metreye kadar bir kıyısal ekosistemden gerçekleşmesi ve biyotop çeşitliliğinin az olmasından dolayı, az bir biyoçeşitlilik olan alanını kapsamasından dolayı olduğunu düşünmekteyiz. Ve yine sonbahar ve kış mevsimini tayin edebildim. Az sayıda birey mevcuttu. Çoğunlukla ilkbahar mevsimi ve yaz mevsiminde daha çok tür artışı görüyoruz. Çalışmam hala devam ettiği için o bulguları maalesef ekleyemedim. Ama tür sayımın az olması bu sebeplerden dolayı. İlerleyen zamanlarda benzer çalışmaların artmasına bağlı olarak ortamda oluşabilecek kuşların tespiti için bu canlı faunasının bilinmesi ve düzenli aralıklarla izleme çalışmalarının yapılması gerekli. İzmir Körfezi zaten sürekli izlenen bir bölge. Ben bu çalışmayla denizel araştırmaların bir bölümünü oluşturmasıyla ve tür çeşitliliği çalışmalarına bir destek niteliğinde olmasını istedim çalışmamın. Sonuç olarak amacım İzmir Körfezi'ndeki mollusк dağılımını belirlemek, bölge hakkında bilgi sahibi olmak, bölgedeki gastropod bivalv türlerini tanıyıp, inceleyip dağılımlarını belirlemek ve tür çeşitliliği, biyoçeşitlilik çalışmalarına destek vererek denizel fauna çalışmalarına katkı sağlamaya çalıştım. İleriki yıllarda farklı örnekleme yöntemleri kullanılarak daha derin kapsamlı istasyonlarda ve derinlikleri fazla olan bölgelerde çalışıldığında daha farklı kayda değer önemli bilgilere de ulaşabileceğini ve var olan eksikliklerin de zaman içinde giderileceğini düşünmekteyim. Kaynaklarım. Dinlediğiniz için çok teşekkürler.
Teşekkür ediyorum Şule Hanım. Sorumuz var mıdır? Sunumunuz için sağ olun. Soru var mıdır? Yok herhalde. Tekrar teşekkür ediyorum. Başarılar diliyorum.
Çok teşekkürler hocam.
Sağ olun. Okay, the last presenter is Aida. And one of my friend will share the video that Aida prepared before. Furkan, could you please share it? Furkan'cığım paylaşabilir misin? Ekranı durdurabilirsiniz.
Evet hocam, bir saniye.
Tamam. Ekranınızı
Ayda mı?
kapatabilirsiniz şöyle hocam. Aida and Professor Ibrahim are here now. If you have any question, please make your notes at the end of the video, at the end of the presentation. You may ask your questions to them. They are here.
Ayda is here.
Yeah, they are here. Aida and Professor Ibrahim are here. If you have any question, please ask your questions
I'm
at the
here.
end of the presentation. Yeah, you are here. Furkan'cığım, tamam. So this presentation was prepared before. Now we can watch it. Falan ses yok Furkan.
Bakıyorum ona. Ben kapattım.
Ses geliyor arkadan. Bu
Ses
kimin
yok mu
sesi?
hoca?
Ses yok, görüntü de yok.
Hocam
Hayır.
ses yok mu?
Hayır.
Autrement, tu peux présenter.
Diğer yüzünden geliyor hocam. Ses de sunum yapacak arkadaş düştü. Yani
Ses
düştü değil.
onun düşmesi problem değil. Biz videoyu oynatabilir miyiz şu anda? O ses nereden geliyor arka plandan?
Good morning. Hello.
Heh.
My name is François Komla Eda. I am from Niger. I am glad to do my presentation on beta globin gene polymorphism and prevalence of plasmodial infection in 3 epidemiological profile of malaria in Niger. It is a project financed by the Pierre Fabre Foundation. And to do our presentation, we made a plan. In our plan, we have the introduction, methodology, results, and at the end, we have the conclusion. Several molecular abnormalities have been highlighted in the beta globin gene, and this gene is located on chromosome 11. There are two genes of beta globin. Changes in this gene can lead to the formation of quantitatively abnormal hemoglobin or to the synthesis of normal hemoglobin in abnormal quantities. Sickle cell disease is a hemoglobinopathy hereditary with autosomal recessive inheritance hemoglobin A mutation. The World Health Organization reports that 38,403 deaths of sickle cell sickle cell anemia in 2019, increase of 26% compared to 2000. In Niger, we have 15 to 25% of sickle cell trait and 1 to 3% of the major sickle cell in some research. The malaria is a vector-borne disease. It is one of those rare public health scourges that has persisted for centuries without diminishing in scale or severity. Sickle cell disease and malaria are both serious illnesses with a similar geographical distribution, particularly in Africa, where limited access to healthcare and socio-economic challenges contribute to their spread. In order to have more statistical data on sickle cell disease and malaria in Niger, our project joined a study of the National Malaria Program Control in their study of mapping of plasmodial species in Niger to carry out our first study on a national scale and according to the epidemiological strata of malaria in Niger. Our main objective in this study is to calculate the prevalence of plasmodial infection in patient care in different sickle cell alleles in 3 regions of Niger. Our study is a descriptive cross-sectional study enrolled patients which blood spot samples were collected across the 3 regions of Niger, including Niamey, Tillabéri, and Agadez. The inclusion criterion were patients with patients favored during consultation in target healthcare facilities during the study, September to November 2022. At the end of this study, we have to collect 5,000 samples on the 50 cities throughout the territory of Niger. But in this presentation, we will present you 1,638 samples that we analyze. Our samples are collected on Whiteman paper, and to do our PCR of threat, we have 3 steps. The first is the DNA extraction with DNA in vitro gene kit. The second is the conventional PCR to amplify our DNA, and the real-time PCR to have our genotype. The threat method uses 2 probes, the red probes and the green probes. When the probes are side by side, excited the donor fluorochrome, which will transmit its energy to the recipient fluorochrome, which will emit fluorescence, which will allow us to have our genotype. These graphs represent the derivation of fluorescence intensity versus time as a function of temperature. For a given allele, each time a TM is researched, dehybridization of the detection probe is observed, which result is a sudden drop in fluorescence and therefore in a sudden increasing of derivation fluorescence according to the temperature. A patient homozygote is considered have 1 peak as genotype of A and genotype S for the allele. And here, when the patient is heterozygote, we have 2 peaks. Here we can see it in green line. In the blue line for the AS, we have 2 peaks, 1 for the E, 1 for the S. And we have to take care to read our result in the range because the reading range is always between 55 and 70. After this range, we having to read the graph, the result. And we have the TM specified for each allele. For the C, the TM is 55 and 57. For the A, it is between 59 and 62. And for the S, it is between 64 and 67. And for identification of plasmodial species, the sample for the identification were on slide, and the reading was done with the thin swirl microscope to have our result. In the result, the first graph is the sickle cell genotype of our sample that we have analyzed on FRIT method. In general, we have 33.6% of polymorphic genotype. In them, we have 29.11% of sickle cell trend and 3.95% of Myers sickle cell syndrome. In our samples that we have analyzed, we have 4 plasmodial species that we found: Plasmodium falciparum, Plasmodium malariae, Plasmodium ovale, and Plasmodium vivax. According to the malaria epidemiological stratum, we have found some forms of infection of Plasmodium. We have infection of Plasmodium falciparum only. We have B infection of Plasmodium falciparum and malariae. We have the infection of Plasmodium malariae only alone. We have infection of Plasmodium ovale and Plasmodium vivax. In low malaria epidemiological stratum, we have found Plasmodium malariae in very loud and moderate. And very low and moderate, we have Plasmodium ovale. But in very low epidemiological stratum, we have found also the B infection of Plasmodium falciparum and malariae. In the moderate epidemiological stratum, we have found Plasmodium ovale. The p-value is 0.7 in the multivariate analysis that show us that there are not a correlation between genotype status and plasmodial species. In our conclusion, sickle cell disease and malaria remain geographically linked and represent major public health problems with reserve a closer thought throughout the development of a better prevention strategy against malaria and better care for sickle cell patients for the greater good of our population. On this, we are at the end of our presentation. Thank you for your kind attention. Goodbye.
Thank you very much to Aida and Professor Ibrahim also. Dear participants, do you have any comment or questions? Professor Ibrahim Karidio is here. Please say hello to us. We cannot hear you now. Your microphone. Okay, now
Hello.
Yeah.
Hello.
Hello. Thank you very much for the
Thank you
nice
for your
presentation.
patience for following this video. Actually, we did that video to prevent any delay that might be due to the internet connection. So that's why we've done this video in advance, and hopefully send it to avoid such inconvenience. Thank you very much.
Thank you very much. I think it's a very good idea to have this kind of presentations also. Since you are here, there is no problem. Sometimes we have very bad time when we use internet. We have difficult time. So, dear participants, if you don't have any question, dear Aida, thank you very much, Aida, for the nice presentation.
Thank you too.
If you have any question, please let me know. Otherwise, we will have a nice photo at the end of the session. Please turn on your camera. One of my friends will take some screenshots to have a memory. To
Of
remember,
course, hocam.
to remember
I
this
take
session.
a screenshot now.
Okay.
Okay. Zufar Alhado.
Alhado,
Can you open your camera?
Vishnu, can you turn on
Okay.
your camera? Alhado, Vishnu, can you hear us? We are waiting for you. Thank you very much.
Thank you, Vishnu.
Vishnu. Professor
Thank-
Ibrahim, we also want to see you there.
Okay, I will take screenshot now. Faruk
Okay.
hocam, is it okay?
Yeah, it's okay.
Okay, Ibrahim is coming. Okay, I will. Okay, thank you.
Thank you very much, everyone. Nice to see you. Nice to meet you in this session. So the next session will start at 15 past 2. Is it correct? So we have a lot of time before the session. Hande, do we close the session now? We cannot hear you. Your microphone.
Yes.
Okay.
And we
you-
Okay. If someone want to say something, we can
We can
stay.
finish.
Otherwise, yes, we can follow your discussion. Otherwise, we will have a break. Okay. Thank you very much, everyone, for the nice
Thank you.
presentations and contributions. See you.
Thank
Bye.
you very much.
Thank
for
you.
Bye-bye.
everything. Bye.
Bye.